We decided to choose prolactin as our molecule because I want to be an OBGYN, so prolactin is relevant to what I'm interested in. And Gabe is a really good friend, so he let me geek out. Prolactin is a protein hormone with 198 amino acids, 23 kilodaltons, as its genetic code. The molecule has 3 disulfide bonds. Prolactin was discovered by Oscar Riddle. He studied the mechanisms for lactation in pigeons and found that the unknown milk-stimulating substance was distinct from other growth-promoting and gonad-stimulating hormones found in the anterior pituitary. Riddle was then able to extract the substance, which he then named prolactin.
Prolactin was originally named the luteotropic hormone because its first function was discovered to be the formation of the corpus luteum in the ovaries of rats. Prolactin is known most for its role in the maintenance of mammary glands and the production of milk. However, prolactin has various functions in different mammals and invertebrates. Prolactin is involved in reproduction through its stimulation for the formation and maintenance of the corpus luteum in rats and mice. Prolactin affects the corpus luteum. Prolactin can either be luteotrophic after mating or luteolytic in the absence of a mating stimulus. The luteotrophic actions of prolactin is characterized by enhanced progesterone secretion, which is essential for the implantation of the fertilized ovum, maintenance of pregnancy, and inhibition of ovulation. Prolactin may be the luteolytic as well by inducing programmed cell death in the corpus luteum. The corpora lutea of the rat has the capacity to express monocyte chemoattractant protein 1, which interacts with prolactin on proestrus to induce luteal cell death.
Prolactin is also involved in growth and development. For example, newts cannot undergo metamorphosis without prolactin. In homeostasis, it maintains water and electrolyte balance. Prolactin osmolarity effects are also seen in minnows, as prolactin allows minnows to adapt to sea and fresh water. Prolactin plays a significant role in regulation of the humoral and cellular immune responses. Prolactin stimulates mitogenesis in both normal T lymphocytes and the NB2 lymphoma cell line. The actions of prolactin within the immune system are initiated by ligand-induced dimerization of its receptor, the PRLR. Prolactin stimulates thymic function and increases the secretion of thymolin, which is a thymic hormone which plays a role in T cell differentiation. Prolactin affects the formation of blood vessels. Angiogenesis is inhibited by proteolytic fragments of prolactin. There are specific high-affinity saturable binding sites for the 16 kilodaltons fragment of prolactin on capillary endothelial cells. There may be a therapeutic use for prolactin fragments as local inhibitors of tumorigenesis. In primates, pituitary and placental growth hormone variants, and the growth hormone-like placental lactogens all bind the PRLR. Hormones that bind the PRLR are collectively termed lactogenic hormones. Prolactin also has synergistic effects on luteinizing hormone receptors and maintains these receptors. This is a view of the female breast and the mammary glands. The mammary glands consist of the lobule, which contain the alveolar cells. Here is a close-up view of what goes on in the alveolar cell in response to prolactin. Prolactin is involved in the production of milk from mammary glands from the following mechanism. A stimulus, such as suckling, will stimulate the paraventricular nucleus to secrete thyroid-stimulating hormone from the hypothalamus. Prolactin is secreted as a prohormone by acetophils, also known as lactotrophic, in the anterior pituitary. The prohormone is then cleaved to become a protein. Prolactin. Prolactin enters the bloodstream and circulates through the body. Prolactin binds to its receptor, PRLR, on the alveolar cells and causes it to dimerize. The tyrokinase JAK will then cross-phosphorylate. The proteins STAT5A and STAT5B are recruited to the complex, where JAK will phosphorylate them. Activated STAT5 and STAT5B are transported into the nucleus and bind to the GAS sequence on gene promoters. This will induce the transcription of target genes that encode proliferation and lactogenesis. Prolactin is a part of the negative feedback loop. The hormone also affects the ovaries, and when there's too much prolactin present in the ovaries will produce inhibin. Inhibin serves as a negative feedback and stimulates the hypothalamus to produce the prolactin inhibiting hormone and also produces the gonadotropin inhibiting hormone. Dopamine is the main prolactin inhibiting factor. Dopamine is secreted by hypothalamic neurons where it enters the blood from the hepatic portal system and binds to receptors on lactotrophins, inhibiting the synthesis of prolactin. Estrogen positively controls prolactin synthesis and secretion. The increasing blood concentrations of estrogen during late pregnancy increase the levels of prolactin to prepare the mammary glands for lactation. Prolactin, however, is stimulated by several hormones such as the thyroid-releasing hormone, gonadotropin-releasing hormone, and vasoactive intestinal peptide. To summarize, these are the inhibitory and excitatory molecules and or stimuli that affect prolactin. Dopamine is inhibitory, while TSH, suckling, and elevated estrogen levels during pregnancy are all excitatory. Interestingly, a baby crying is enough of a stimuli to prepare a mother for lactation.
Here's an overview look to see how prolactin is regulated by the hypothalamus and pituitary glands. Here we have the hypothalamus, which contains a group of cell bodies called the arcuate nucleus, and they produce prolactin inhibiting hormone, also known as dopamine. Dopamine then travels down the hypophyseal portal system, where it gets secreted into the anterior pituitary. Once inside the anterior pituitary, the dopamine binds to receptors on the lactotrope, which then inhibits the production of prolactin. We also have in the hypothalamus paraventricular nucleus, which secretes thyrotropin-releasing hormone. TRH also travels down the hypophyseal portal system and enters the anterior pituitary. Once inside the anterior pituitary, it also binds to receptors on the lactotrope and stimulates the production of prolactin. Estrogen can also stimulate the production of prolactin. When females are pregnant, they produce estrogen, and when there are high concentrations of estrogen present, it can actually directly stimulate the lactotrope and producing prolactin. Estrogen can also come up into the hypothalamus and inhibit the arcuate nucleus from producing dopamine, which then results in the higher production of prolactin. Prolactin affects mammary gland development. Disrupting the prolactin gene or knockout of the prolactin receptor results in abnormal mammary gland development, which is characterized by complete absence of labio-alveolar units in homozygous females. Impairment of mammogenesis in homozygous mice for prolactin knockout and prolactin receptor knockout results in mice being unable to produce milk. Prolactin R in the mammary gland is phosphorylated when prolactin binds and activates the JAK2 and STAT5 pathway. This pathway is responsible for both mammogenesis and lactogenesis. STAT5 is activated by the long form of prolactin R, which induces transcription of milk proteins. Lacking functional genes encoding the prolactin receptors prolactin, prolactin receptor, STAT5A, and STAT5B are essential in the role of mammary gland development and or reproductive function. STAT5A deficient mice result in females that are unable to lactate after childbirth because of a failure of terminal differentiation. STAT5B deficient mice result in impaired mammary gland development. Milk proteins are expressed in the STAT5B deficient mice, but there is insufficient milk in order to feed the pups. Females that have only one intact prolactin receptor allele are unable to lactate during their first pregnancy, but during their second pregnancy they are able to lactate due to the stimulation of progesterone and estrogen that compensate for the loss of the prolactin receptor allele. When a normal protein-producing cell in the pituitary gland develops a mutation, it becomes prolactinoma. These pituitary tumors will continuously divide, making more prolactin-producing cells and resulting in the overproduction of prolactin. Prolactinoma can be found in men and women, but is more common in women. Symptoms in women include irregular or lack of menstrual cycle, infertility, menopausal symptoms, or milk discharge from breasts. Since dopamine is the main inhibitor of prolactin production and secretion, it can be used to treat prolactinoma. Medications used as dopamine agonists include bromocriptine and cabergoline. However, an excess of dopamine results in the underproduction of prolactin, also known as hypoprolactinemia. Hyperprolactinemia is a result of pituitary underactivity which results in a decreased level of prolactin circulating in the blood. Other sources for prolactin deficiency can be due to familial puerperal alactogenesis, traumatic brain injury, or circulatory diseases. Puerperal alactogenesis is hereditary and results in the inability to breastfeed. Symptoms of hyperprolactinemia can include menstrual disorders, delayed puberty, infertility, and decreased milk production. Treatments of hyperprolactinemia can be achieved by taking drugs that act as dopamine antagonists and remove the inhibition of the lactotrope cells.